Now, for a dive into sexual reproduction in humans specifically, the syllabus asks us to really look at the male reproductive system first. So, we're going to be looking at that right now. Okay? So, the male reproductive system, we're going to be looking at its structure, its physiology, and its anatomy. Alright? So, it's generally just anatomy and physiology for all of this. Right? So, the male reproductive system, it should be quite familiar for some members of the population. For all members of the population, I'm not sure. Right? But it should be very familiar. Alright? So we know that we have the different structures that we look at. I'm going to go through the structures, right? We have the testes, which are encased within the scrotum, right? Specifically, which hangs outside the body because of suitable temperature, right? The sperm occurs at a lower temperature than body temperature, therefore it hangs outside the body. We should know that, right? That's a general fact. Then we're gonna have our epididymis, then to our vas deferens, passing our seminal vesicles through, right? Well, a little bit under our prostate gland, right? And then through the urethra, right? To reach the penis specifically, right? And then we're gonna also have the bladder and the ureter in this system, right? But we're not gonna speak about the bladder and the ureter a whole lot, right? We're just gonna talk about the reproductive capabilities of this system, right? Because in males, it's combined. The urinary system and the reproductive system are combined. We're just gonna look at the reproductive part of this system, all right? So if any questions come up, just let me know. So the testes specifically, we know that testes serves as the primary male reproductive organ. These are the gonads, right? So we're looking at this now. So it's responsible for what we call spermatogenesis, right? You know what spermatogenesis is? It is the process where we have spermatozoa being produced in our seminary first tubules. All right. The testes themselves, right, as gonads play a key role in hormonal control, right? So a hormone crucial for development of secondary sexual characteristics, right? Testosterone is mainly produced in this organ, right? Which cells produce testosterone? Please remind me. Which cells? We don't remember which cells? Okay, I want somebody to type it in the chat which cell produces testosterone, whether it's the serotonin or the Leydig, right? So we're gonna look at those. Right, those things can come in any way, shape, or form. So the structure of the testes now are highly convoluted tubules. So their tubules are really highly convoluted. That's really their superstructure of just completely binding tubules, right? So highly convoluted tubules maximize the surface area availability, right, for sperm production or spermatogenesis, ensuring a plentiful supply for fertilization. So we know that each minute goes past, we create millions upon millions of sperm, right? If we are a member of this Google meeting that possesses testes, right? A lot of it is produced, and it is produced until death, right? So from birth going until you die. Technically, it continues to produce after you die until all the cells stop working. But yeah, so we have that there. Right, we have the epididymis, which is this structure that is nestled right on top of our testes. Right, the epididymis is a tightly coiled tube located on the posterior surface, right, of of each testis. Right, so we have each testis, many testes, each testis. Right, so epididymis is found on a testis. Right, so its primary function is to store and transport spermatozoa produced in the testes, right? Additionally, allows for the maturation and acquisition of motility by sperm cells, right? So we know that our spermatozoa, right, or the singular spermatozoon is motile, right? So it gains its motility while it is there being going through maturation in our epididymis or in the epididymis, right? So the structure of the epididymis, with its convoluted tubules and microvilli lining walls, provides an extensive surface area for sperm maturation and interaction with secretions that enhance motility and viability. In some males now, these secretions are, you don't secrete a lot of these solutions, right? And in some males, they have a lack of the secretion of these solutions, therefore their sperm are not viable. So they will produce sperm from now till kingdom come, but no fertilization, no proper fertilization will take place because they are not viable. All right. So in some cases you can have that. All right. So vas deferens, or vas deferens is what is coming out of our epididymis, or of the epididymis, right? So the vas deferens, also known as the ductus deferens, is a muscular tube that transports mature sperm cells from the epididymis to the ejaculatory duct during ejaculation, right? So structurally, it is lined with smooth muscle tissues which contract and propel sperm forward through peristaltic movements. So you remember peristalsis, right? The contracting and relaxation of muscles, right, along different tubes in the body. For example, you know that your esophagus goes through peristaltic movements as well, right? So this muscular structure enables for the vas deferens to efficiently transport sperm over considerable distances, right, ensuring their um delivery to the reproductive tract during sexual intercourse, right? So we have that there. Everything is fine there. Everything good so far. We did the testes, the epididymis, and the vas deferens. Lovely. The seminal vesicle now. The seminal vesicles are glandular structures located near the base of the bladder, right? And they produce significant portions of seminal fluid, semen, right? So semen is generally a milky precipitate, well, not precipitate, it's milky secretion, right? That nourishes and protects sperm cells. So this fluid contains fructose, right? And it contains some amount of calcium ions. It contains a lot. It's basically a nutritious like mixture, right? That is meant for the sperm cells, right? So it provides energy for sperm motility as well as prostaglandins, right? And prostaglandins help to facilitate sperm movement in the female reproductive tract, right? So the structure of the seminal vesicle with their convoluted tubules and high vascularized walls optimize for the secretion and delivery of seminal fluid during ejaculation. So these are the seminal vesicles here, these huge structures, and this is nestled right under our well, right above our prostate and right under the bladder, right, and the urethra is right there going through. So we have that there. The ampulla. The ampulla is basically an increase in a little bit of vascularization, right, within our vas deferens before we enter the seminal vesicle. Right, that's really it is. And the isthmus is just the where the vas deferens meets the vesicle. All right. Well, it's not. Yeah, it's generally that area, that rich area that is found on the prostate that it goes under. Okay. So the prostate gland, is a walnut-sized gland, right? This is some very American units of description. Usually you'll say it's whatever centimeters high and whatever circumference. We're gonna use an Americanized system and say it's walnut-sized, right? So it's a walnut-sized gland, right? Situated just below the bladder and supporting the urethra. So the urethra is actually snuggled there, right? A milky secretion it produces a milky secretion that forms a significant portion of semen, right? So semen comes from the seminal vesicles, as we stated before, right? So it makes a huge amount of semen, right? A huge proportion, right? And the prostatic fluid contains enzymes and citric acid, right, which helps to activate sperm, right, and enhance motility. So notice that almost all of the fluids that we talk about so far enhance motility, because the spermatozoa have to get somewhere, right? It has to enhance motility, right? The structure of the prostate gland with its lobular arrangement of granular tissue and smooth surface muscles allows for the secretion and expulsion of prostatic fluid into the urethra during ejaculation. All right. So it has muscular tissue at the top, so it's going to squeeze, right, that mixture of enzymes and citric acid into the urethra. Right. So we have that there. And we have the bladder there. Okay. The bladder, and we have the vas deferens there, or well, the urethra technically, and then we have the prostate. Okay. By the way, just mentioning, I just keep on mentioning facts on each slide, but like we know that prostate cancer generally tends to be high in men. It tends to develop around the age of around late 30s to early 40s. They are around there. Um, there are specific there are specific molecules within tomato plants and other plants that are similar to tomato that actually can inhibit the issues that actually cause prostate cancer. So if you do not eat tomatoes, then it may have an early onset. It's not a will, but it's a may. Okay, so you know, healthy eating is very important. Are you the urethra now? The urethra is a tube that serves as a dual function in males, right? Acting as a passageway for both urine and semen. So it extends from the bladder to the tip of the penis, where it opens right to the exterior, to the world itself, right? Structurally, the male urethra is divided into three regions: the prostatic urethra, the membranous urethra, and the spongy or the penile urethra. The urethra has muscular walls and mucous membrane lining facilitates the passage of urine and semen during urination and ejaculation, respectively. So the blue is here, right? What's happening here? And then the blue would be the prostatic, right? The green here would be the membranous, and then we have the spongy going all the way down. All right. The penis specifically now. So the penis is a male organ of copulation and urination specifically. How we know it has a dual purpose. Structurally, it consists of three cylindrical bodies of erectile tissue, right? Two corpora cavernosa, right? And one corpus spongiosum, right? During sexual arousal, these tissues fill with blood, causing the penis to become erect, enabling penetration during intercourse, right? The urethra runs through the corpus spongiosum and opens at the tip of the penis, allowing the passageway for semen during ejaculation, right? Additionally, the penis is richly um, innervated, right? Contributing to sexual pleasure and orgasms, right? So what we're saying is that it's innervated, meaning that it has tons of nerves. It doesn't have the same amount of nerves as your eyes, but it acts as a ton of nerves, right? So we have the general structure of the general area there. Okay. All right. So that should be fine. That should be lovely. Okay. Any questions, concerns thus far?
Sir, can you go back over the episode that part, please?
The epi,
Um,
the epi what?
yeah,
The epi what?
that word.
The epi?
So,
Oh,
sir,
epi,
sir, you were
epididymis?
talking about. Yes, yes, sir. That.
The epididymis. All right. So the epididymis is just really some. It's really just a collection of convoluted tubules, right? That are right on the testis, right? So basically, what happens there is that when the testis produces the sperm, right, it stays in the seminal vessel. It stays in the epididymis, right? So when the male is not ejaculating, this is where the sperm is stored. So the sperm is stored in the epididymis. So if you ask for a function there, the sperm is stored there. Sperm mature there as well, right? And they are mixed with different types of secretions that help in their maturation specifically, right? So they're mixed in these secretions that help with the motility and viability of the sperm. That's fine.
Okay. Yes,
Any
sir.
okay, lovely. Any questions for anyone else? All right. Spermatogenesis now, the origin of spermatozoa. Oh, somebody said 71st tubules 7 minutes ago. Oh, that was 11 minutes ago, so probably wasn't in reference to asking a question. Okay, so spermatogenesis now. We know how it is because spermato and genesis. Genesis is the creation of something, and sperm. Mato would speak to the spermatozoa. So spermatozoa being created, spermatogenesis. Alright? So an overview now, we know that testes are primary male reproductive organs are structurally composed of several key components. While each testis, right? So within each testis, right, there are numerous coils, right? Numerous coils. So these coils are called seminiferous tubules. Alright? So we have the seminiferous tubules in it, right? Within it. Okay? And then what we have now, these tubes are now lined with two types of cells. We have the serotonin, right? And the spermatogenic cells, right? Or we call the Leydig cells. So the serotonin cells provide structural and metabolic support to developing sperm cells, right? And are involved in the regulation of spermatogenesis. So spermatogenic cells, now Leydig cells, right? Generally help within, well, spermatogenic cells are not Leydig cells. Hold on, there's a difference. I'm gonna explain the difference when we reach there. Actually, they're not. Right, so spermatogenic cells are where we get our is a part of the basal layer. I'm gonna explain that, right? So the spermatogenic cells now, right, are allows for the cells, right, to go through it allows, it's the origin of spermatogenesis. Let me say that, right? So surrounding the seminiferous tubules now are interstitial layers of cells are Leydig cells. So I was mixing it up a while ago. Alright. So the spermatogenic cells are a part of the basal layer, and as you go outside the basal layer, now there is where you're gonna have your Leydig cells. Yeah? And your Leydig cells are the ones that actually create the testosterone. Alright? So this hormone, the testosterone, is important for the development of secondary sexual characteristics and the maintenance of male reproductive function. Right? And we know what secondary sexual characteristics are for males and females. Right? We know the increase in hair, the broadening of the shoulders, those type of stuff that you will see. Right? Testosterone can increase that. Right? So we have this now. So what we have the epididymis out here, so but at the testes now we're gonna have the seminiferous tubules, right? And we're gonna have the tunica albuginea, then the tunica vaginalis, then the cavity of the tunica vaginalis, right? What that is describing, right, is generally layers that it is held in, right? Layers that the seminiferous tubules are held in, and together is called the testis, right? All right, proliferation now. We're going to go through spermatogenesis now. Proliferation, or what we call mitosis. Spermatogenesis begins with the division of the spermatogonia, right? Which are diploid stem cells located along the periphery of the seminiferous tubules. During this phase, spermatogonia undergo mitosis to produce two types of cells. Type A spermatogonia, which remain as stem cells, and type B spermatogonia, which differentiate into primary spermatocytes. Alright? Now the primary spermatocytes now that comes from the type B spermatogonia, right, are diploid, right? So what's gonna happen here now, they're diploid, so they're gonna resulting well, they're diploid, and that what it's saying is that it's resulting from the mitotic division that undergoes in meiosis 1, right? Or that happened before, and then they continue to undergo meiosis 1, and this process results in the formation of secondary spermatocytes, which are haploid cells containing half the number of chromosomes as the original spermatogonium, right? So originally, the spermatogonium would have gone through mitosis and creates primary spermatocytes, right? The primary spermatocyte will undergo meiosis 1 to create secondary spermatocytes, right? Each secondary spermatocyte now will undergo meiosis 2, resulting in the formation of spermatids. Meiosis 2 separates 6 chromatids, producing 4 haploid, right, cells from each primary spermatocyte, right? Then we have spermiogenesis. Spermatids undergo spermiogenesis, a process of cellular remodeling during which they differentiate into spermatozoa, right? So we have that. So spermatids become spermatozoa through spermiogenesis. Some textbooks or some books will say that this is called specialization, but it's the same thing, but the scientific word for it is spermiogenesis, right? This involves extensive structural changes, including the formation of the head, which contains the nucleus, the midpiece, which contains the mitochondria, right, and the tail, which is the flagellum, right, for motility. And then, last but not least, we have spermitation. Once matured, spermatozoa are released from Sertoli cells into the lumen of some of the first tubules, and that is called spermitation. So the release of them, right, from spermatozoa was the release of a spermatozoa, right, from Sertoli cells into the some of the first tubules, right. That is what spermitation is, right. So the first thing we know that is proliferation via mitosis to produce the different spermatogonia from one um, original primordial cell, right? Then the primary spermatocyte that comes from the mitosis of the type B spermatogonia, right? It goes through meiosis one to produce a secondary spermatocyte. Then after secondary spermatocyte goes through meiosis two, it becomes a spermatid. And then the spermatid goes through spermiogenesis to become a spermatozoa. And then the spermatozoa are released from the Sertoli cells into the lumen of the seminiferous tubule through the process called spermitation. That is what's happening there. Okay? So this is spermatogenesis here. So we have spermatocytogenesis, spermatidogenesis, and spermiogenesis. All right, lovely. So we have the primordial germ cell, right? That will divide. Let me make sure that you guys see what is happening now. That will go through mitosis producing cell type A, right? Remember that type A won't do anything. It will return up here. Type A will just remain as the primordial germ cell, while type B, right, now goes through the first goes through cell DNA replication, right, and splits, right? So this is the first meiosis, right? So meiosis one to produce secondary. So let me put the one with the degree here to show that this is a primary spermatocyte. Then we're gonna have two secondary spermatocytes on the first round of meiosis, right? And then it's going to then what we're gonna have now, right, is wait, hold on. Did I look at something? But give me a second. I'm labeling the thing in the wrong area. Nobody can tell me that I'm labeling it in the wrong area. Oh my. So we have after we have mitosis, we're going to have the creation of the spermatocytes. Hold on. It's because I draw the line short up here, right? So we have that there, right? The spermatogonium will actually go through some DNA replication, then create this now. So it's gonna go through the first meiosis here, right? To get our primary spermatocytes. Second round of meiosis to get our secondary spermatocyte. Spermatocyte right there. And then what we're gonna have here now is become our spermatids. So these are now our spermatids. And our spermatids will now mature into spermatozoa. Right? So we're looking at here now. Here we have the Sertoli cells. I love how this connects everything, right? So it connects the entire diagram to what's happening here, right? So here we have our basal layer. So this is the layer that I call the basal layer, or that we should call the basal layer, right? And up on the basal layer here, we're going to have the different spermatogonium. The spermatogonium will become a primary spermatocyte. So note that it pushes down to get a primary spermatocyte, right? And then from there now we're going to have the secondary spermatocytes that is produced. And then when those finish now, they're going to become a spermatid, and it's going to completely mature now, right? So when they're mature, they're gonna start looking like this. So this maturation, right? And then then spermitation occurs when they are ejected, right? And they just now flow freely in the lumen towards the epididymis to be stored, right? That's what's happening. So the Sertoli cells are there, right? And the Leydig cells are also there, all right? So we need to understand now what happens, right? What we do to really promote spermatogenesis, right? It's really a negative feedback, right? So our hypothalamus will create GnRH, which is gonadotrophin, right? And then that will stimulate our pituitary gland to produce FSH, which causes our serotonin cells to produce inhibin, right? And it produces LH, luteinizing hormone, which causes our Leydig cells to produce testosterone, right? So when our Leydig cells produce more testosterone, right, or when there is a lot of testosterone produced, right, we know now, right, the body will know that there's a lot of testosterone produced, right? And that will signal the serotonin cells to produce even more inhibin, and the inhibin will stop the pituitary gland, right, from actually producing more FSH and more LH that will produce more testosterone, right? So when testosterone is shown or found, right, when it's at low levels, we're gonna have gonadotrophin by the hypothalamus, right, which is our master, which is there, right, and our pituitary gland, which is our master gland, right. So we're gonna get gonadotrophin that's gonna cause the pituitary to produce FSH and LH, right. Serotonin cells produce inhibin, Leydig cells produce testosterone, right. And you guys should be able to understand this diagram if it were ever to become, well, if it were ever to come rather. All right. So it's just a feedback mechanism for the hormonal control of spermatogenesis. So we have too much testosterone, then that will decrease the production of FSH and LH based on the diagram. All right. Any questions or concerns as it relates to spermatogenesis, right, or its hormonal control?
Sir, I don't understand.
Okay, lovely. So we know that we don't understand, but where don't we understand?
The diagram. I don't understand the diagram.
Oh, this diagram here, the hormonal control.
Yes, sir.
All right, so let's try to put it into a stepwise approach. Let's look at it. All right. So let's start from the hypothalamus then. All right. So our hypothalamus produces gonadotrophin. All right. So our hypothalamus, let's say that it exists by itself, it produces gonadotrophin. All right. That's the first step. Produces gonadotrophin, right? Gonadotrophin causes our pituitary gland to produce FSH and LH. That's the second step, right? Right? Second step. But this is a negative feedback loop. That means too much of something will cause everything else to turn off, right? So we're gonna have FSH. FSH stimulates our serotonin cells. Follicle stimulating hormone will actually do that. And our serotonin cells, as you should remember, are the cells, right, that our spermatids are embedded in. So we're gonna need that to produce spermatozoa, right? And LH now, right, will actually cause our Leydig cells to be more active, which produces testosterone for sexual maturation, right? And some amount of nutrients, right? So we have that there. So these two cells are important for spermatogenesis, right? But what it's saying now is that testosterone, however, right? Testosterone, however, actually stimulates serotonin cells to produce something that we call inhibin. Inhibin inhibits the pituitary gland from producing FSH and LH. So you'd understand now that once we have a lot of gonadotrophin, right, we're gonna have a production of a lot of testosterone, and then as we increase our levels of testosterone, the serotonin cells will start to produce inhibin, which turns off our pituitary gland. It tells our pituitary gland to say stop producing FSH and LH, right? But testosterone also acts indirectly though. So it actually acts indirectly to tell the pituitary gland to stop, right? And also, right, it tells our hypothalamus to stop gonadotrophin production, right? So that is what it's really doing. Does that make a little bit more sense more that I go know that I go over it, or is it that you need more time to look at it? I'm not sure. Is that better?
A question,
Go ahead.
sir. So you're saying that is it that because of like increased levels of testosterone production, and then you have the pituitary glands stimulating FSH to stimulate the Sertoli cells to release inhibin. That's a negative feedback.
Yeah, so it's a negative feedback. So as we increase our products, right? Oh my gosh, not I was about to make that sound so much like chemistry a while ago. But as we increase our amount of hormones, the hormones that we tend to increase in the levels of actually causes the production of inhibin. So it kind of is a negative feedback loop, actually. So if the male body or the female body, because females do produce testosterone, just like how males do produce estrogen, right? If we have too low levels of testosterone, there's not gonna be enough inhibin to prevent the pituitary gland from producing more testosterone. So if you think about it like that, it's a cycle. We know homeostasis. We understand homeostasis from the last part of cystic biology, right? That with something like osmoregulation or hormonal regulation, right? If we have too much of something, our body is going to turn off the production of that thing. So basically, that's what's happening. Is that fine, Damien? Okay, lovely. Are there any other questions? No? All right. So this is another diagram looking at spermatogenesis. So remember, this is the Sertoli cells. This is all happening on the Sertoli cells, while the Leydig cells are producing a little bit of nutrients for this to happen, and it's also producing testosterone. So this is another way at looking at it, and this breaks it down. I love this. In the basal layer, we have proliferation through mitosis happening. Mitosis happens twice, right? First time from the spermatogonium to produce type A and type B spermatogonia. Type A goes back to replace the original spermatogonia, right? You know why? Because if this both spermatogonia was used, then the um then what we're gonna have is that spermatogenesis will occur and it will stop completely forever. Right? So we need that first spermatogonia from mitosis to return there to continue producing more B-type spermatogonia. And the B-type spermatogonia now will go for another round of mitosis, right? And this is now getting closer and closer to the lumen, right? So here we pass the blood testes barrier, right? And now we're in the adluminal barrier, right? The abdominal area of the seminiferous tubule, right? So what's happening here now is spermatidogenesis, right? Where we're gonna have the primary spermatocyte go through meiosis to produce our secondary spermatocyte, which actually go finish meiosis two, right? So remember, meiosis happens in two steps. So meiosis one, then we have meiosis two. Meiosis one of a primary gives us a secondary, and meiosis two of a secondary gives us a spermatid. Now the spermatids go through elongation and maturation, right? Which will actually be finished, right? And after it's finished, we have spermitation, where they're now ejected as spermatozoa into the lumen. And this differentiation here, some textbooks love to call it specialization, or they love to call spermiogenesis specialization. All right? So please remember that. All right? So we have that there. I remember all of this is occurring in the seminiferous tubule, right? Spermacytogenesis, spermatidogenesis, and spermiogenesis all occur in Sertoli cells, right? A part of the Sertoli cell, and then when the Sertoli cell ejects them, now that is permutation into our lumen. Okay, lovely. So notice as the process comes now, we're going closer and closer to the lumen. All right. Now the structure of the spermatozoa. Right. An overview. Look at the overview now. That's an overview of the structure. Should make sense. Or corona and sagittal view. But we're gonna go into it, right? So the head of the sperm contains a nucleus, which houses the genetic material necessary for fertilization. The head is covered rather by a cap-like structure called the acrosome, which contains enzymes essential for the penetration of the protective layer during the surrounding the egg, right, during fertilization, right? Remember that layer rather, that protective layer that is around the egg is called the corona radiata. So it will actually um going to produce enzymes along with hyaluronic acid to actually dissolve that layer. We're going to talk about that in fertilization. So we have the head there, right? The mid piece located behind the head contains numerous mitochondria arranged in a helical pattern. These mitochondria provide energy in the form of ATP, as we know, for the movement of the flagellum, enabling motility. Right, so that's where the flagellum, right, or generally the flagella, gets the energy from for its rotation. Remember, it rotates, you know. It's a corkscrew turning, it rotates. And then some spermatozoa have two flagella, some have three, right? And they start to rotate and bumping each other, right? So it's motile, right? So the flagellum is a long whip-like structure extending from the midpiece, and it's composed of microtubules in a 9 plus 4 pattern, a 9 to 4 pattern, right? Characteristic of flagella in eukaryotic cells, right? The coordinated movement of these microtubules allows the sperm to swim through the female reproductive tract towards the ova for fertilization, right? So we have that there. So the flagellum, okay? I made special note to mention the acrosomal vesicle as a part of it. The acrosomal vesicle there because we need to make sure that zona pellucida and the corona radiata is completely removed. Well, not completely removed, but penetrated in order for fertilization. But I just made note of the acrosomal vesicle here, or what we call the acrosome. All right. So relating its structure to its function, we know the acrosome is a specialized vesicle that contains the enzymes and hydrolytic enzymes as well as acid to actually pierce the zona pellucida and the corona radiata, right, and move through the corona radiata, right? We have the head, which contains the genetic information for fertilization. We have the midpiece that contains mitochondria for the helical movement. Well, not for the helical movement. In a helical pattern, right? For the movement of the flagellum, and we know that the flagellum enables the sperm's motility, right? And that is it for this presentation. We did the male reproductive system as well as how the gametes looks, the structure of the gametes. All right. Any questions or concerns?